This is Jason Key at SB Grid at Harvard Medical School in Boston. So thanks for joining us today. Today we have Abhishek Singharoy. So he's joining us from the Theoretical and Computational Biophysics Group in Champaign, Urbana. Abhishek is going to talk to us today about flexible fitting, molecular dynamics, flexible fittings of X-ray diffraction data, though I understand that his own work is focused on large-scale simulations of organelles. Go ahead, Abhishek.
Okay, yeah, thank you. Thanks for the opportunity and thanks for spending this time with me. Let's get started then. So structural data is actually available at different spatial scales. So this slide kind of summarizes the amount of data that is actually available. So are the movies playing well? Are they looking smooth enough?
They look good from here.
Okay, good. So cool. What you see here on this slide is the type of experimental methods that are available, the data types that those methods provide, and the let's say the length scale, the spatial scale at which this data, which captures this data. So we have the NMR and the X-ray crystallography data, which gives you very nice idea about the all atom resolution of a structure, the secondary structure, the tertiary structure. Then obviously you go on to the cryo-EM and cryo-ET, where you start getting idea about the overall organization of a system. Nowadays, obviously with cryo-EM, we are getting information not only about the overall organization, but also about the positioning of the side chains, a lot more detailed information, but still cryo-EM and cryo-ET gives you scales that are much larger at this point of time in comparison to X-ray crystallography. And then finally, you will go on to other types of experiments like FRET experiments, which would in fact give you cell scale information, like how is a cell dividing. So this is the more or less the amount of structural data that is available to us, or at the scales. So these are the scales at which we can actually obtain, we can see a structure form and a structure change. So obviously, understanding a structure is very important for the functions itself. So if you are interested in X-ray crystallography, often you are also interested in things like how is the protein functioning. I've given an example of a membrane permeation process. So generally, X-ray crystallographic data or NMR data would end up transcending into discoveries in membrane permeation. Similarly, when you go on to cryo-EM, you'd be gaining more knowledge about motor action mechanisms. You'll be getting knowledge about molecular movements. And similarly, at the cell scale, you'll be getting knowledge about things like growth, cell division. So these are various different scientific targets that are kind of aligned with the structural assignments that people use, starting with these sort of data types. So now the aim of the work that I'm going to show today is to interpret this data across multiple scales in all atom resolution. And so these are the tools that we have been developing in Urbana-Champaign, where what we do is we start using molecular dynamics simulations, and we integrate data from different experimental sources coming from different lengths and time scales, and try to interpret that at an all atom level. So this can be the structure of a protein, the structure of a complex, or even the structure of a cellular organelle. As I move on, I'll show you that later on in the slide. So that's the aim. So basically what we want to do is we want to model cell scale objects at all atom resolution by integrating data from different experimental sources. So with that then, let's move on to the next slide. So here I introduce this idea of molecular dynamic simulations. So the overall idea of the processes that we are dealing with is the fact that molecular dynamic simulations provide a very nice platform for integrating multiple data types. So first, let me introduce the molecular dynamic simulation methodology a bit, and then we'll get into how do we, in fact, incorporate experimental data types into the molecular dynamic simulations. So again, what is molecular dynamic simulation? So a molecule can be analyzed at different scales. At an atomic scale, if you see a molecule, then you can make an assumption that a molecule is nothing but a bunch of atoms that are connected by springs. Each springs have a separate spring constant. So under that assumption, what one can do is one can write these potential energy functions. What these potential energy functions are, they are some combinations of energies that looks like springs. So you have bond energies, you have energies for angles, you have energies for dihedrals, you have energies for electrostatic interactions and van der Waals interactions. So what you do is then you take your molecule that's composed of a bunch of atoms, and the atoms are interacting using these potentials, this U of R potential. Uh, then you use you solve basically Newton's equations of motion. And the idea is that a molecule would change its shape or size such that its overall energy goes down to a minima, because the native state of a molecule is in fact its lowest energy state. And so the equations here would are solved in such a way that the molecule evolves so that it reaches a certain equilibrium stage. I'll give you an example here. Let me give an example of a protein folding problem. So we start out with so this is the native state of a protein, which is folded. So we start out with a highly unfolded state of a protein, and this molecular dynamic simulation, which is like solving the Newton's equation, evolves this structure of a coil to something that is more structured, which is the native form of the protein, which is in fact also the lowest energy state of the protein. So this is so in fact now you understand that the only data that this sort of simulations need is basically the potentials, the potential functions, the fact that there are atoms and they are bound by strings. So if there is no structure at all, then this tool by itself is good enough to provide you structure from literally a coil. But that's not always the case, obviously, and therefore we need experimental data guidance in order to obtain structures, in addition to whatever the force fields of a molecular dynamic simulation can provide. So that takes us to the next step, which is this idea of a molecular dynamics flexible fitting. So normally, what we have is we have data in the form of electron density, data in the form of an envelope. Let's say if so, what one can do is one can use this electron densities that, for example, comes from X-ray crystallography or from cryo-EM, and then construct a potential. We call that UEM, uh, which let's not get into the formulation of that unless someone is interested in it. So what one can do is one can take this electron density, one can write a Coulomb-like potential term out of that electron density and add that onto the force field. This UMD is the force field that I introduced in the previous slide. So now you have a new force field that includes in it the terms from the original molecular dynamics force field, which will always allow your structure to conform to a native conformation, to conform to a low energy conformation. In addition, you have a term that is coming from the experimental data. Let's assume that it's electron density data, and so now what this force field would do is it would generate structures for you that not only gives you low energy, it would also give you structures that conform to the experimental data that you have. And in addition, there are certain restraints that one needs to add to it that I'll introduce a bit later. So again, so now you have a new potential energy function, and this function can now, with this new function, you can again start solving the Newton's equations. And what you'll see is on the right, you start out with a structure that is not fitted to a density, and then gradually the forces that are coming from the density are actually driving the structure into the density, and you get a fitted structure. So this is the overall philosophy of the methods that I'm going to introduce here onwards. With that, then let's move on to the first type of methods, which is the electron density that is being obtained from crystallographic data, so X-ray diffraction data. So for this, we have a tool that we called xMDFF. Uh, so let me introduce the tool a bit. So what this tool does is you have your diffraction data. So you start out with the diffraction data, and you start out with an initial model. So the initial model can be a homology model, for example, and then you combine the initial phasing model with the data to get an initial electron density. And then to this electron density, you fit this initial model to get a new model. Let me show that in a movie. And that new model, once it comes out, you can ask the question, is the R value greater than a tolerance or lesser than a tolerance? If it's lesser than a tolerance, then it's your final model, else you continue this iteration. So in a movie, it'll look like this. So a density, so it's basically a very small fragment of a protein that I'm showing here. So what I'm showing you is that the density is evolving within iterations. So each change of density is actually an iteration. And so the density is evolving, and finally, and the protein is kind of getting fit to the density till finally the protein would in fact get fit. and you will get a final solution. This is the local change. And you can also see a global change for this. So the orange structure is an initial guess that you have plugged into this simulation. And this is a test case. So you know what the final structure is. It's going to be the CN structure. And xMDFF, in fact, starts doing the calculations, and it starts refining the CN structure. Sorry, the CN structure was the initial, and the target structure is orange. So xMDFF starts with the CN structure, brings about deformations in it, and then finally, so that the structure becomes the red structure, which is the known target structure. So what we're doing, in other words, is we are conforming the structure to the available diffraction data using molecular dynamic simulations through the use of extended force fields. So that's the philosophy. So now with this kind of technique, one can go in and start doing some test cases. So what we did was we picked up some low resolution cases from the protein data bank, and and then we started refining them. And what you see is that we managed to get better results for these set of 6 proteins, uh, both in terms of mole probability scores and as well as R work and R free. So mole probability is something that is always that always becomes very good in a molecular dynamics simulation framework, because the force fields are always very good in terms of giving you your side chains and backbone geometries. So therefore, you can always take it for granted that the mole probability score is going to be really, really nice. And the other thing that we also see is that the R work and the R free, the difference between those two has come down, which means that the amount of overfitting is not as much. It's in fact lower than whatever people had reported back in the day. With that, then we moved on to okay. Here is another very interesting facet of this kind of simulations. Often there's a question of how do you treat disulfide bonds in doing your refinements, and therefore in this particular when you're doing it using xMDFF, you actually have the liberty of choosing places where you want to insert a disulfide linkage and where you don't want to, and then you finally see what is its impact on the R free, on the R factors of the refinement. So here, for example, I started out. So this is a complex called cobratoxin. So we started out with an initial structure that was already submitted in the protein data bank, and then we ran Phoenix on it, and we got reasonably good results. Uh, then we redid xMDFF once with by considering that all the disulfide bonds are reduced, so there is no disulfide linkage, and in the other time where all of them were oxidized. And then what we found was that indeed for the oxidized case, we get much better refinement statistics than for the reduced, which is even better than what is submitted or after 24 cycles of Phoenix. So this gives us the feeling that we can actually temper around with the chemical nature of a problem and see under what condition or under what chemical condition is our result fitting the best to the experimental data. So xMDFF gives us this sort of flexibility in terms of uh, testing the chemical correctness of the model along with the fact that whether it's fitting the data or not. So then these we started doing other applications which were totally de novo applications in terms of these were our collaborators with whom we worked, and we started refining structures. So there are at least 5 or 6 structures that have now in fact come out. They are already submitted to the protein data bank. I'll show you an example of one particular refinement. So this is a refinement of a voltage sensor protein. This is in fact the biggest protein system that we have done using xMDFF. And what you will see is that we start out with a structure that is fairly deviated from what the final structure was. That also gives you an idea of the radius of convergence of xMDFF. So you can really start out with structures that are quite deviated from what the final structure is going to look like, and then xMDFF is going to give you the final structure. So here you can see that the R free and the R work and the mole probability scores, these were all pretty high to start out with. And then as xMDFF happened, so xMDFF in fact started with a very medium confidence model that was predicted by mufold, a very medium confidence homology model. And then we managed to get down to uh, R work and R free of 0.26 and 0.28 respectively, starting with something that was 0.45 and 0.47. And our mole probability score also went down. And then finally, these helices, the placement of these helices are very important because it's a voltage sensor domain, and you really need to know where the helices are. If they are either one turn low or one turn up, then it's a different functional state altogether. So we wanted to understand what was the actual functional state of the protein under the data uncertainty of 4 angstroms, and which xMDFF actually managed to resolve and show that this was indeed the ground state of the protein, and not the excited state, or not the uh, charged state of the protein, where the helix is like one turn up or one turn low. So it managed to, in other words, it managed to resolve the interhelix counters even at data uncertainty of 4 angstrom, which was something amazing to see. Uh, then, so xMDFF, okay, xMDFF allows another thing. So because xMDFF depends on force fields, so you can also resolve abiological problems with xMDFF. You don't have to have biological, like you're not necessarily restricted to solving biological structures. So here's a structure of a macromolecule. It's called a Rotaxane. It's a molecular motor. It's like this molecules like this has been given the Nobel Prize in this year's chemistry. Like, so the Nobel 2016 chemistry came in Rotaxanes. So if you want to get the structure of a Rotaxane, and if you want to do something like Phoenix, you would see that the structure is pretty distorted and you're not getting the right phasing. Another reason for that is because the underlying structural information that Phoenix derives is primarily for biomolecules. However, if you give it a new molecule, then it doesn't have enough information for to resolve the structure of that molecule. But xMDFF uses force fields, so therefore we can in fact resolve abiological structures. So what I show you on the lower left is the, let me go back. Is the fact that there is a huge amount of structural phase space that one can resolve with xMDFF. So the initial structure is on the extreme left, uh, and then xMDFF did a huge amount of phasing in the translational and the rotational space. So it sampled all the space to finally come and settle down on a structure that is the final structure shown in green. And the zoom out, you see that it has now been able to resolve the density very nicely. And there are multiple occupancies, so you can in fact see multiple stereoisomers of the same system that was embedded within the crystallographic data. So with this, then, and it's purely because xMDFF can manage to dynamically sample multiple states of a structure, as well as it is also empowered by very good force fields. So putting these two things together, we can in fact start learning, start even doing phasing with xMDFF. But that's only for small molecules. So normally people would use something like ShellX to do these sort of problems. However, as the size of the abiological macromolecules are growing, ShellX is becoming more and more. It's getting very difficult to handle this sort of problems with ShellX. So people in this small molecule community are in fact looking for macromolecular crystallographers to come solve their problems, because whole molecule disorder is something that we know how to do really, really well. And But we don't have good force fields. So what xMDFF does is it basically combines the forces of the macromolecular community with force fields in order to solve problems also in the abiological community. So that is xMDFF for you, and the tool is available here. You can download the tool. There are tutorials, and then you can also always. So we are always a very proactive group in terms of helping people with ways of solving the problem. So before I move on to my next data analysis, if there are any questions, I'd be very happy to answer. That would also make the thing a bit more interactive. So yeah. So
Yep. If there are any questions, you can send them by chat. You can send them to me, and I can pass them on. I had a couple of questions. So the xMDFF is doing refinement in real space. Is that correct?
correct. That's correct. That's absolutely correct. So it's a
And
real space refinement technology. Yes.
during the process, is it cycling? Is it iterating back and forth using Phoenix to regenerate maps, improve?
Yes, indeed, indeed. So
Yep.
if I go back quickly to this, so yes, we are dependent. So it is using Phoenix only to the extent that it needs a new electron density map every iteration. Having said that, so therefore, it's very simple to couple Phoenix with xMDFF, because on the step where we are actually generating, so it's particularly here in the step where we're actually regenerating a map, we can also run a little Phoenix refinement and then regenerate a map. So you can run an xMDFF refinement and a Phoenix refinement back to back without any problem in the same protocol, and that's a part of the tool anyways. So therefore therefore you will need Phoenix, and at this point you can use Phoenix only for map generation, or you can use Phoenix for refinement while running xMDFF. So then you can kind of double dip. You can use both the tools simultaneously.
So that way you would get a reciprocal space refinement, and then
As well as a real space refinement. Yes.
Yeah, and Phoenix now, I think it's in the latest nightly, also has its own real space refinement. So
It does. It does.
yeah, so you could
Yeah.
conceivably look at that at some stage too.
Absolutely. And in fact, so one of the in fact, if you think about it, historically speaking, there was real space refinement in Charm Explore. So which was the way I think the difference is the radius of convergence. So basically, the way in which you use your real space refinement affects the radius of convergence of your tool. So because we do it using very soft potential, so that would be a very different kind of an argument. But let me so you can so think of it. If you have an equation and you're targeting the equation based on a number, you're simply telling me that change my position so that my R factor goes down. It becomes a very stiff constraint. However, in xMDFF, we have much softer constraints. So it allows us to make much more, sample a lot more structures. I think, and that's why we have much larger radius of convergence. So when you say that Phoenix has a real space refinement tool, I completely agree with you, and I know I'm very aware of those tools. And I think the difference between, and in fact, a Phoenix real space refinement within our real space refinement would in fact make the tool a lot more, a lot more powerful. But the idea is that the basic difference is the radius of convergence. So we indeed provide a lot more radius of convergence purely because of the fact that we're using molecular dynamics, which Phoenix has not yet been able to do.
Yeah.
And therefore, these are very complementary. So we just go hand in hand here.
I've got a question here from Pete Meyer.
Mm-hmm.
So
Yeah.
for low resolution X-ray maps, I'm curious to know, have you tried that with experimentally phased electron density in addition to model phased electron density from Phoenix Refine?
Yes, absolutely. So this particular example, we did both. So because that was the first case where we not only wanted to solve a structure but also wanted to compare it with experimental phase density, we exactly did that. So, and they matched really, really well. So therefore, we submitted two structures, one that came out with xMDFF and another that came out with experimental phase refinements. Yes. So
Do you have to remember which type of experimental phasing was used there?
I think it was MAD. I think they had used MAD.
Okay, thank you.
Yeah.
Right. Yep. Great. If you want to go ahead with the next section, I think that would be
okay. Cool. So now then, in fact, there was one more example. Not only this, so there is also a very recent structure paper where SAD was used, in fact, and so there are at least two examples. So let me just quickly show Pete these two references. So there is a NSMB citation and there is a new structure citation, which is there on the slides. You can see that in both the cases, it in fact we did that. So just to make sure that things are okay. Okay. So now then, yeah. Now then, let's move on to cryo-EM. And so, okay, again, I had introduced the theory a bit earlier because that was also the same piece of information that went into xMDFF. So now let's quickly move on to what is this risk. So what is this USS? That was a point that I had left out the last time. So both during the molecular dynamics flexible fitting simulation, which is where you take a structure and conform that structure with respect to a density, and xMDFF, where the density also changes, we can use several restraints. Some of the restraints being secondary structure restraints. For example, you really don't want to overfit. You really don't want to break a helix when it's at or a beta sheet, let's say, when it is being fitted to a density. So you can put in those restraints using again different kind of spring constants that holds those helices together for you, and those are already implemented in MDFF. There are things like domain restraints where you really don't want to do too much. Like suppose your data is very low resolution and simply want to fit an entire domain within that density. It's pretty similar to, so it would be like rigid body with some more sampling to it. So you can like take an entire domain and can so plug it into a density, uh, constraining it to the fact that its overall shape and size is not going to change. Not only can you do that, you can use symmetry. Suppose you have like things like a virus, for example, where you know that you have the repetitive copy of the same protein. What you can do is you can end up fitting one protein to the density while using symmetry information, so that you can just copy over that protein to 10 other places in the density, as we show here. So basically, in the fitting on the lower, extreme lower right, only one protein was fitted to the density, while symmetry information was used to regenerate the protein in all the other subunits while running MDFF. So basically, you're actually saving your compute time by using symmetry information. So these are things that are already available on MDFF. Uh, you can do very large structural changes in MDFF now. So what I'm showing you here is you can take a certain cryo-EM density and you can make low resolution equivalents of that and start fitting with the lower resolution density, and then gradually improve the resolution to fit to very high resolution density. So often what happens is, because this is a molecular dynamics based simulation tool, molecular dynamics is notorious for getting trapped in local minima, and MDFF is no different. It gets trapped in local minima, especially when the density is very well resolved, very highly resolved. In order to overcome that problem, we have come up with this idea of cascade molecular dynamics, where that was the movie that you just saw right now. The fact that you start out by a density that is washed out, it has very little information, it has just the information on the overall architecture, and then you gradually refine this density. So this is a tool that really allows us to give provides us very again high radius of convergence. In fact, we are showing that using high resolution density as an example. You can do this for any density, and in fact, we have seen is that for any density at any resolution, a treatment like this often gives you much better results than a direct molecular dynamics flexible fitting. When I say direct, it's just take the structure, plug it into the density. Rather than doing that, if you do a bit of density refinement together with the molecular dynamics, then you end up getting much better structures. So this work has been reported now. So, and in fact, this is the only way perhaps in which MDFF can resolve high resolution data. Uh, Next, okay, and obviously, when you have high resolution data, you would want to see if your finally the side chains, are they in fact how are they being resolved, are they being properly fitted to the density. And what you see here is this, for this particular example of the TRIPv1 channel Yifan Chang's data, you're actually seeing that the xMDFF results are not only giving you a very nice overall architecture, that's the background, but they're also giving you very nice side chain alignments. And this process is very hands off. You just start your simulation, start your refinement, and then you would see, like, this thing works like a magic bullet, in fact. So we have seen very little problem with this particular protocol. So this perhaps is the best MDFF protocol we have at this point of time for fitting something. Next is something we also wanted to give the user an option of working interactively. It's like, for example, like Coot. Just there are a couple of differences. The fact is that you are actually solving Newton's equations of motion while you are actually pulling or pushing a piece of protein inside density. Basically, this prevents you from doing something really, really unphysical, and your geometry still remain good. So what you see here is that we're interactively placing a protein inside a density. The arrow that you saw was where force was applied to the protein, and you see that an MDFF is normally happening, and now together with the MDFF, you're using your own intuition to fit a structure to the density. And then on the lower right hand corner, you're seeing that the local cross correlation is actually increasing, which is fine. And the other very important thing, what this interactive MDFF allows you to do is the fact that it, in fact, much unlike Coot, where it's a very local endeavor, where you have to do changes segment by segment. Here you can actually see an allosteric effect. Like, you just push one part and you see that the entire protein is kind of coming towards that particular direction. It's purely because, again, a molecular dynamic simulation is running in the background. So you don't have to worry about the global changes, provided you know how the local changes are. You have good intuition about the local changes. So if you induce a local change by hand, manually, interactively, then the global change will be taken care of by the fitting procedure, by the underlying molecular dynamic simulation. This, we feel, is perhaps the way to go as we move forward. And so therefore, again, there's a very nice plugin on this tool called VMD, Visual Molecular Dynamics. So this which allows you to run, which is, so this Visual Molecular Dynamics is, let's say, it's like the chimera to MDFF. So it's a visual tool where it's a visualization tool that allows you to see what is going on inside molecular dynamics flexible fitting. And so there's I would heavily encourage people to have a look at this particular tool, because I find that this will be very helpful. You can use your intuition when in doubt, but at the same time you can also use all the goodness of molecular dynamic simulations in making sure that the geometries and the energetics of the problem are all nicely taken care of all together, all simultaneously. Okay, so uh, so let's talk a bit more about model validation. So, but there is a huge amount of discussion on in the field of cryo-EM. What are the good criteria for model validation? There are 6 different ways in which molecular dynamics flexible fitting can actually validate model, which is in fact reported in the eLife paper that I'll show, which I showed in a couple of slides back. I'll just take one particular error analysis criteria, which we found was very interesting, and as well as very physically intuitive. So there is this concept of a local resolution of a model, or local resolution of a map. So what we found was the local resolution of a density, in fact, can be directly computed from the amount of fluctuations that MDFF that one can compute from MDFF. So if you see any one of, I'm just going back here just to show you what I'm talking about. So if you see that while an MDFF simulation happens, the protein in fact is fluctuating. You see the fluctuations in this particular movie. You have seen fluctuations in all the movie that I've shown you. So what I'm trying to say is, if you compute the amount of fluctuation of these proteins, then that fluctuation tells you the quality of the map within which the protein is fit. So if the map is lower quality, if it's lower resolution, then the local fluctuations of the fitted protein inside the map would in fact be higher. So in fact, there is a direct relationship, a linear relationship between the local fluctuations of a fitted protein and the quality of the map locally. So with that in fact, we can predict local resolution of a map from MDFF, and that is what we are showing here. So we have we have colored a protein by local resolution, and we have colored the same protein by RMSF, and we see that it is I think on the upper part we have so we have 2 different type of proteins here. We on the upper row is beta galactosidase, and the lower row is TRPV1, and you see that the local resolution, uh, coloring by local resolution, which is the left hand, is very similar to coloring by RMSF square, which is and the square of your RMSF gives you beta factors. So you can literally compute beta factors from an MDFF calculation, which will help you sharpen your map. You can compute the local resolution of the density from MDFF, which will allow you to kind of segment your map based on what is the local resolution. And so this is one very important, and this comes in very handy for high resolution maps, in fact, where you have very high resolution regions and very low and also lower resolution regions. So you can actually segment your map based on MDFF results by tracking this quantity called root mean square fluctuation, which is also now, so this analysis is also now integrated into into the software called VMD, which I showed in the last slide. So therefore, MDFF provides a very nice way of 6 different ways of validating maps. I'm showing you 2 of them. In fact, one of them, which is through this RMSF, which tells you that based on MDFF results, you can right away predict the local resolution as well as the beta factors. So again, there are other ways of getting. There are more established way of getting beta factors, and we wanted to see how does the MDFF beta factors, which is shown in black, tally with the more conventional way of finding beta factors through the Garnier analysis, and we see very good agreement. In fact, these 2 data sets. So the red one is so both this data set is for TRPV1. So the red one is using Garnier analysis, and the black one is using MDFF, and you see that there is a very nice correlation between these 2 data sets. So what then this means is that MDFF, if you take an unsharpened map, do a fitting with MDFF, from there get RMSFs, then you can right away get beta factors, which you can use to sharpen the map. So MDFF is also providing you a tool of not only fitting the map, but also refining the map further by computing beta factors on the fly. So that is MDFF. And now this is all the associated software that's available. That's first of all, so in order to run MDFF, we would require a tool called NAMD, which is a molecular dynamics program, a tool called VMD, which is a visualization program. So these tools should be, and you'll also need Phoenix. So in order to run both MDFF and xMDFF, so okay, just for MDFF we'll need VMD and NAMD. For xMDFF we'll need VMD, NAMD, and Phoenix. That all those three things you'll need to be on your laptop or on your computer. And then you have to generate input files. So nowadays we have a very nice website which in fact generates the input file for you. So what you do is you go to this website called charmgui.org, input MDFF, or there's an xMDFF version to that, and you upload your diffraction data file or your cryo-EM data file, and then and the initial model, the PDB structure of the initial model that you want, and then you keep hitting some buttons on that website. What you'll see is that the website will start generating files that are required to run MDFF and xMDFF. So the output of that particular the output on that website will be a folder that contains all the necessary input files. So you don't have to do anything by basically you don't have to if you don't know what molecular dynamic simulations are, you don't really have to learn that to run MDFF and xMDFF, because that's what we are doing in the back end. And once you have the files ready, you can just put one command line and then one command line argument, like you have to just say namd2 and the name of the input file, and the refinement will start running for you. So we also have tutorials. And so with this, I think I would conclude the MDFF part of my talk. And if there is any question on that, let's address that before I move on to the third segment, where I'll start showing you how do we use these techniques uh, uh, to kind of set up cell scale models, really large structures, which are much bigger than just proteins or protein complexes. So let's take any questions if we have on the MDFF.
I just note that NAMD, VMD, and Phoenix are all available in the SPGrid install. So those are all there together. One question I had while we're on the specifics of the software itself, what are the computational requirements for a standard refinement if you expressed it in something like CPU hours per number of atoms or something? How long should the user expect to, do you need a cluster? Can you run
Right.
a
I would say uh, for the systems that I have shown so far, you would not need a cluster. You might even end up doing that on a laptop. So I would say a laptop to a standard workstation. So when I say a standard workstation, I would mean I'm not even expecting GPUs. I'm expecting like a quad core CPU, something like that. That would in fact be appropriate for getting these. So the compute requirement is not as much. So you don't need a cluster, or you don't need um, let's say a supercomputer to run the examples if they are like proteins or simple protein complexes. However, yeah, and in fact that was one of the reason to use ideas. If you have like the if you can manage to bring in the element of symmetry in the problem, then that really brings down the the amount of compute cost for you. And a lot of the times that is that in fact is true, that you can utilize symmetry uh, to, uh, to refine your data. But even if you can't, I think from all the standard complexes that we are seeing right now that are coming up, they can always be done on a laptop to a CPU, to let's say to like a quad core Intel quad core machine, let's say. uh, so they're not very compute intensive. But again, people, suppose you want to do something like a proteasome, which is like getting into bigger protein complexes there. uh, you would in fact like to get into clusters. But the other problem though is, once you're getting into bigger computers, you're losing your capability of interacting with the things. So I think this slide number 18, which is this interactive MDFF, which is a very important tool, I believe. So I think one should do these sort of simulations as interactively as possible, and to make that happen, you in fact resort to CPUs. So yeah.
Is there a GPU acceleration component to MDFF? I know
Yes,
that.
yes. So the GPU acceleration component to MDFF comes in from the GPU acceleration component of NAMD. So basically, it's NAMD that's running on the back end, and NAMD is heavily parallelized and and it has like, if you use GPUs, it's highly GPU compatible. So if you run this using GPUs, that makes life so much faster. Like the movie that I showed you here, like the interactive part was on a CPU. On a GPU, you would have seen this happening 10 times faster at least. So there's a direct efficiency improvement on GPUs, absolutely. Uh,
Good to
and
know.
VMD, and because VMD is also available on GPUs, so I would say simple CPUs or GPUs would be the best way to go if you want to do things interactively and do it for, I would say, protein complexes to the to I don't know, let me put in a number here, like 30,000, 40,000 atoms we're talking about, we are good within on a computer. So on a laptop to on a workbench, on a work machine, where we don't need supercomputers. The next thing that I'm going to show you will actually, in fact, need supercomputers, but then I will tell you why is that the reason.
All right, great. Well, why don't we jump ahead to that? Take a
Uh-huh.
look. Yeah, go right ahead.
Okay, cool. So now then, so far I had been talking about doing data separately, like separately do your crystallographic refinements, separately do your cryo EM refinements. How come? But there are situations that arise, but you have multiple data known for the same system. So you have, for example, in this particular case of when we were getting the structure of a protein called HITC. It's a ribosomal insertase protein. So what the function of this protein is, just for the sake of the biology, is like what it does is, so ribosome is this huge molecular machine that synthesizes protein for us, and then these protein HITC, it's an insertase protein. It sits right at the bottom of the ribosome. What it does is it takes the protein from the ribosome and inserts it into the cell membrane. So here is where a cell membrane is, and so this thing comes in and take the protein from inside the ribosome and either drops it inside the cell membrane. Assume that there's a cell membrane here, sorry, the cytoplasm here, or there's a cell membrane on the either side. So it inserts it uh, into the cell membrane or into the cytoplasm. So in order to refine this model, we have three different pieces of data. We had data from on the ribosome from cryo-EM, that was fine. We had some parts of the proteins that were known from crystallography within the ribosome, cool. And we had some parts of the protein that were not known. So what we had to do was we had to come up with this evolutionary covariance analysis, which in fact gave us an idea of how is the secondary structure of the protein, how did it co-evolve, so how close are the helices to each other. So this is an analysis that is very hot nowadays, where people are going back to the evolutionary history of a protein and coming up with what is the overall arrangement of the proteins with respect to each other, or of the, so let's, of the secondary structure components with respect to each other. And so that is this particular figure here, but you in fact see that we have additional restraints that are coming in the forms of distance restraints from evolutionary covariance. So we have also information on the fact that there are certain residues that should be within a certain amount of distance from each other. So then this is an additional restraint that we can add on to our simulation. So we have restraint from the electron density, we have restraint from the crystallographic diffraction data, and we have also restraint from the evolutionary covariance. And then you put all the three restraints and you do it and you do MDFF. So so now also for this particular problem, you need a supercomputer purely because of the size of the problem. Because you have to deal with the entire ribosome while we're also fitting the protein. So you in fact have a density for the entire region, for the entire system here. And so this is where you cannot in fact use local compute resources, because it is going to break your computer, your local computer, or even a very cheap cluster. So you at least need a GPU cluster, let's say a 64 core cluster, to do these sort of calculations, where you are not only fitting with multiple diverse, at least three different kind of data sets, but also your system size is increased to the level of ribosome, which is like a full a full organelle, so to say, a full machinery. So that's why it is important, so that we can run these sort of tools in a supercomputer. And however, the problem is, as I said before, you lose that level of interaction that you in fact want to have. However, so far it had turned out well. Like even without interacting with this, with this refinement on the fly, you saw that MDFF did a good job. But there is always the fact that you can always get data out of the supercomputer, do some local treatment and put it back. It's a bit more tedious, but you can also do that. So one of the after finding after this result was we published this result in terms of finding the structure of this little protein here called HIDC. A crystal structure came out. So we found a result based on cryo-EM and based on the distance restraints from evolutionary covariance. But then later on, a crystal structure came out, and then sorry. What you would see is that the 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 crystal structure is the one in blue and our structure is the one in red. So the overall three-dimensional arrangement of the proteins was very nice. uh, However, there was some differences in the packing, which was purely because the crystal structure was not obtained with the ribosome present. The crystal structure was obtained in isolation. In the presence of the ribosome, the protein changes its tertiary structure a bit, which we managed to capture, and there's a biological reason for that. But the good thing was, starting from something like an so in fact, like, it was an starting the initial model was completely guessed here. So it was like an evolutionary covariance model on which we only had some information about certain distances, and we had a map that was also not a very high resolution map. But when you put these two things together, the structure that came out was very reasonable and was very agreeable with the structure that came out later from a crystallographic study, which also adds to the confidence of this sort of hybrid modeling that one can do. Now then, I think, yeah. Then we tested MDFF on cloud computing resources. This was on the Amazon Web Services, and we saw that if you see, so again, like the performance and the CPU necessity, all those things are on this table. You can see that amount of time that is required. So this is data for the. So this particular data is for. Let me go back a bit, and I'll show you the exact protein. So this particular data was for this protein, the protein on the lower end, strip V1. And also for so these two proteins, if you see these two proteins, and now these were like 4 subunit proteins, around 30,000 40,000 atoms that needed to be fitted into the density. And this is a pretty high resolution density, and so this is the compute performance that we are talking about. So in fact it costs lesser than lesser than a cup of coffee, depending on the brand, obviously. But yeah, so you would see that it's very inexpensive compute wise. And so when I say 5 nanosecond a day, so in it takes around 2 nanoseconds to fit a structure. So we are talking about much more. So we have kind of made sure the structure is really really fitted, and it doesn't take too long. Takes only less than an hour. Costs like 84 cents. So basically, the other idea is the reason of making a cloud computing compatible version of MDFF and xMDFF is the fact that experimental groups don't necessarily need to have heavy duty uh, theorists in their own group. They can literally use the cloud computing resources. The same philosophy on which, to a certain extent, SBGrid is also built on. Like, it's providing software solutions. So this is a way of us providing the solutions through the cloud computing. Okay, now then, so that was the third segment, and now then quickly let's move on to the fourth segment of, unless there's any question till now, then I will now move on to a bit more biological mode. So this was mostly all the technological software oriented things that I wanted to say today. If there's any question up till now, I'll be happy to answer, else we'll move on. So okay, we're good then. Then, because there is no other question, then let's move on to. Okay, so what do we do with all these tools? So the first thing that we can do is, there is this. So what is the scientific question that we can ask with these tools? So there is. We can start constructing models for cell organelles. So what I show you in front of you is an electron tomography image of bacteria. It's called Rhodobacter sphaeroides, and within this tomography image uh, is embedded an organelle, which is a photosynthetic organelle that absorbs. It's called the chromatophore. It absorbs sunlight and makes ATP. It's a model system for understanding photosynthesis, for understanding the ideas of cooperativity, repair, assembly, stress response on photosynthesis. And so we wanted to construct this organelle in all atom details using data from different experimental sources, and then start asking some scientific questions. So I won't be having time, and today is also not the best of day to talk about the scientific questions, but I would show you how did we make this model. So let me quickly show you. So this particular model is it will also, so the initial idea of where the model, how the model should look like, came in from the cryo-electron tomography experiment. So, came in from the cryo-electron tomography experiment. The first thing we realized was that this particular object, this chromatophore, should be spherical in nature. So it's otherwise it won't fit the cryo-electron tomography density. So that was the first piece of information that we that was obtained from this data, that it's a spherical object, nothing more than that. And then obviously this model was built, so it has like 101 proteins, 16,000 lipids. There are different types of proteins that are embedded in this particular model. So the question is, how did we make this model? So that's what I will now quickly show in the next 2 slides. So the first thing that we did is we had experimental data. So I'll reuse some of the older slides, some of the older pictures. So we had xMDFF that with which we managed to. So the first problem was, how do we get the protein structure? So we used xMDFF to refine the X-ray crystallographic data to get the local, basically what is the secondary structure of the protein. And then there were for some of these proteins there was cryo-EM available, so we could have packed in the protein into a density and get like what is the overall tertiary structure of that particular protein. Then, however, so far we haven't talked about the environment. A lot of the times the structure of a protein depends on the environment it is in. And if you see for this particular organelle, the proteins are embedded in lipid membranes. So one has to now take into account within the refinement the effect of the environment around it. So we took this particular protein that came out of MDFF and redid an MDFF in explicit membrane. That's what you see on the lower right. And the first thing you see is that the protein folds, the protein curves in the presence of the membrane, which is very important, because you think of it, this protein is a part of a sphere. So locally, the environment of the sphere should be curved. It shouldn't be flat. However, in the vacuum refinements that we did so far, it was flat because there was no environment present. Now, in the presence of that environment, the lipid environment, you see that the protein indeed induces a curvature. And then you do a bit of molecular dynamic simulation to make sure that the protein is stable in this curved form, and the curvature is stable. And once you have that, then you plug this information into this sphere, into this spherical model. So you do this 4 different times for 4 different kind of proteins. So 4 different proteins, we have to do 4 different sets of xMDFF, MDFF, a membrane MDFF, and then plug it into the overall model. Fine. So that only gives us a structure of the proteins. We have no experimental information on how are the proteins arranged relative to each other. Where do we get that information from? So in order to get that information, let me stop this movie for some time. Okay. So we had also information from other sources. So after getting the structure from MDFF and xMDFF, which gave us the structure of individual proteins and their local environments, we still needed to know how are these proteins arranged next to each other. So we had AFM data, which gave us a fair amount of idea as to how are the proteins arranged next to each other and what is the distance between these 2 proteins. Those information were plugged into this model. Then we had mass spectrometry information as to what is the ratio of the protein to lipids and how many proteins do we actually have to put into this model. There was also proteomics data which told us how much what is the number of protein components present, what is the relative number of proteins in comparison to the amount of lipids. So it was a huge endeavor in terms of a huge amount of data integration went into it. But all of it happened through molecular dynamics. All of it happened through NAMD and VMD and MDFF. So I had shown you already the use of density restraints and distance restraints, and the same went in. So crystallography and basically electron microscopy data was used using MDFF, and then there were distance restraints that were used, which I also showed for the AFM data, and this mass spectrometry and proteomics, they showed up purely in terms of they contributed to the number of components that are present in the model. And then we constructed this model using using MDFF, as I showed in the last slide. And then you simulate it. You want to see that if this is stable or not. So you're seeing now for the first time a full cell organelle, atom by atom details. This is the first, in fact, the first all atom simulation of a cell organelle. And you see this is pretty stable. You can see the various components, and like there's nothing, this thing is not bursting out. And so now this thing is, it's pretty stable. So you actually end up constructing an entire cell organelle in all atom details by combining all these hybrid simulation tools, which I think should be where, things are going. Um, So we shouldn't just restrict ourselves to just modeling with respect to one or two kind of data, but we want to integrate all these various kinds of data, obviously physically in the right way, on the right softwares, using the right software protocol, and then you get this full atom description of organelles with which you can start doing some scientific work, which is beyond the scope of today's talk. So I will not get into that. So I think this is where I want to stop in terms of the modeling that I have done so far. And then I would like to acknowledge my advisor Klaus Schulten and the various developers in the group who had been working with me in constructing in the towards the software end as well as towards the scientific end of this project of constructing huge cell organelles and doing the developments that sustain this scientific challenge. And then we have again the tools are available on the website. Do help, and if you have do go there. It's a pretty user friendly software. We have a huge user base, so we want to sustain that and continue growing at that user base. So if you have use our software and if you have any questions, always there, email us, and then there's a mailing list on which emails can be forwarded. So and we always respond and make sure that the users are having good experience with the software. So I think with that, I'd like to end my talk today and take questions. Yeah, thank you.
Great. Thank you very much, Abhishek. Questions? So I think we covered the questions as they came up through. If anyone has any questions, you can send them to me by chat, but it seems like things are pretty quiet. So thank you very much for the great workflow of xMDFF, MDFF, and there's an exciting look into what you're doing with it. So it's very impressive scale of simulation, I have to say. So.
Yeah, thank you. I think that's the one reason of using these sort of tools is because the scale is you manage to get into scales that cover scales starting from the angstrom up to several nanometers. We're trying to get to the micron scale. So at the end of the day, one would like to see, let's say, a whole cell membrane in all atom details using all the experimental data available. So that's what molecular dynamics or a tool like NAMD provides you, is the scalability of 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 using all the best computers in the world. But at the same time, we stay focused on the accuracy of the models that we have based on whatever we know so far. So therefore, we not only work on the more conventional crystallography and cryo-EM side of things, but also the more unconventional, let's say, cell scale organelles that you won't be able to see. This sort of modeling you won't be able to see in other groups, purely because there is not enough resource that they want to spend on these sort of endeavors. But if you have the right kind of experimental data, the other thing is also, like, if you think of experimental data, it's coming in from different experimental groups. So it's very important to have this source of data coming in from different groups. And once you have that, then you can use this molecular dynamics as a platform to integrate all those 7-8 kind of data. So that's what MDFF allows you to do. So
Great.
It can allow you to. Yeah.
Thanks again, and thank you everyone for joining, and have a good day.
Yeah, you do. Bye-bye.